Hello everyone, dear ladies and gentlemen. It's a pleasure for me welcome you in this nice venue and start our conference, which is this year in a little bit different format than we are used to. So basically 4 different conferences are joined into the one. So this year we have a privilege to welcome here also the clinicians, not only people from the experimental field of the oncology, but we have here pathologists, and I hope, despite the fact there are more rooms which are focused more clinically than our section, that also some people from the clinical side are here and maybe can somehow give us new ideas, new view on our experimental field. I have to apologize, Marian Hajduch, he will have welcome speech later, I hope, because now he has a meeting with some people from the government regarding the COVID situation and some preventive measures, and what's definitely will be informed later what's preparing and will happen. I would like also to emphasize that the safety is important. So please be careful in the case of any signs of the infection disease or any symptoms. Please be safe and behave safe, and also use all the protective measures at the venue, which was announced upfront. I hope we can start with the science. We are here for the science to discuss our results, and it's a real privilege nowadays, especially that we can join together, not only online but also here on site, and we can discuss our results and we can present our results in the present format. Maybe some people are joined also online, so I would like also welcome them. And if they have any questions, they can send it through the chat, and we will and colleagues will try to answer them. Okay, so I think that's all on the beginning, and I would like to welcome here Zbigniew Nový with his interesting talk, which is focused on the alpha therapy in prostate cancer. Zbigniew, stage is yours.
Thank you for the kind introduction. So good afternoon, ladies and gentlemen. I would like to present you some results from our recent project, which is dealing with PSMA alpha therapy. At the very beginning, I would like to mention that this project is in fact collaboration between our institute here in Olomouc and colleagues in Dresden in Germany. Our colleagues in Germany, they are chemists who are synthesizing these PSMA derivatives. They label them with actinium-225, and they are sending it to Olomouc to our institute for biological testing, mainly meaning in vivo testing. On the introduction, I would like to point out what it is PSMA. In fact, it's prostate-specific membrane antigen. As you maybe know, its official name is glutamate peroxidase carboxypeptidase 2. And to be more confusing, it's called in every other scientific field with another name. So the neuroscientists know it as nalodase, biochemists know it as folate hydrolase, and oncologists use this name PSMA. In fact, it is a zinc metal enzyme which resides in the cell membranes, as you can see on the picture here. And it's expressed in various tissues, mainly in prostate epithelium, proximal tubulus of kidneys, in small intestine, and in ganglia of nervous system. So in some 5 or 6 different tissues, but what is the most important, it is 10 times higher expressed in prostate cancer compared to normal status. So it could be used as a marker of the progression of prostate cancer, and as you can see, it correlates with the tumor progression and also with the relapse of the patients. So it is clear it is a good target for PSM for prostate cancer therapy and also diagnosis. In nuclear medicine, we use both modalities, diagnostic and therapeutic variant, depending on the nuclide which we use for the radiolabeling of this PSMA inhibitor or ligand of PSMA. To show you briefly a current situation in the field of labeled PSMA, we have basically 3 different PSMA derivatives which are already in the clinics or clinical testing, let's say. The first and oldest one is the PSMA 11, labeled as gallium-68. It is a purely diagnostic nuclide which is used for positron emission tomography imaging of prostate cancer. It was already last year approved by FDA for this use. So it should be soon. It is already in clinics for maybe 2 years. The therapeutic variant labeled with lutetium-177 is now maybe waiting for the FDA approval because this year it passed successfully phase 3 of clinical trial. It's labeled with a therapeutic nuclide, of course, with a relatively long half-life, and it has a range in tissue about 2 millimeters, so it is good for treating metastasis and also the primary tumor, of course. What is interesting, this compound was firstly published by Martina Benešová, which is a young Czech scientist working in Heidelberg in Germany. So she is credited for, let's say, discovering this compound, which is now very successful in clinics, clinical testing. It was acquired by Novartis, so it will be soon used as a clinical drug by Novartis. And the latest molecule is labeled as actinium-225. Actinium is an alpha emitter, so it has much larger linear energy transfer. This value says us how much energy it possesses on its way, this particle on its way in the tissue. So the higher number is the bigger is the damage in the tissue. As you can see, when you compare it with the beta emitter, it's much larger. But on the contrary, it has lower range in tissue. This actinium-225 alpha particles emitted by this actinium are able only to reach 50 micrometers in the tissue, which is good for treating micrometastasis mainly. But of course, it can be used in other cases too. So generally said, higher therapeutic efficiency of alpha therapy is clearly visible compared to the beta one. And this compound has started clinical trial phase 1 this year already. Just briefly, one clinical example, nice images with the lutetium labeled PSMA 1617. These are 8 patients, different 8 patients with prostate cancer, and on every patient is represented by 2 PET scans, left and right, in every case. And the red dots are presenting metastasis in the bones mainly and lymph nodes. And you can see the effect of therapy after 3 months with this compound. That clearly on the right ones, right PET scans, there is almost no positivity. So really, really successful therapy after 3 months. So now to our own work in our institute, we are testing new inhibitors of PSMA. They are derived mainly from PSMA 1617, but they are improved in their structure in 2 ways. The first way is using new chelator, which is called macropa. This kind of chelator is enabling us easier and faster actinium labeling. And the second feature is employing so-called albumin binder, so the moiety of the molecule which is binding to human plasma albumin. And this binding should increase the circulation time in blood, so it should enable us to increase the accumulation of the compound in the tumor. We have tested these 4 different compounds, which are named after the chemist, after Falco Isaac. The first 2 are modified with macropachylator, and they have no albumin binder, and the later 2 ones are also with a macropachylator, but enhanced with albumin binder. Here we see the structures. It's maybe better to understand the issue. Here on the left, you can see the macropachylator with the typical nest, which is using for chelating actinium-225. Then we have quite a long linker, which is linking between the chelator and the PSMA binding moiety. FR55 is just a dimer of this structure. The dimerization should lead to the increase in the accumulation in the target tissue. And here on the right, you can see the albumin binding moiety on the molecule. So basically, these molecules are the same with the exception of this moiety. And again, this is the dimerization of the previous one. Very briefly about our methodology, we are using SCID mice in our ex vivo biodistribution study. These mice are injected with LNCAP cells, which are prostate cancer PSMA positive cells derived from the human tumor. We inject 10 millions of these cells into the flank of these mice, and after some 6 weeks, we have the tumors which are big enough to be enrolled into the study. So when the study starts, we apply 50 kilobecquerels of the actinium-labeled PSMA into the mice. Then we sacrifice the mice in these time points. To see the biodistribution, we are dissecting the mice into these 12 organs, which are measured for the radioactivity. And then we count percentage of injected dose per gram of every organ to be able to compare the biodistribution in different organs. And finally, we are sending tumors, livers, and kidneys to our colleagues in the Department of Pathology to do the histological examination. Our histologists, they examine necrosis by hematoxylin eosin staining. They examine also the PSMA expression in the tumors. Also, they watch for DNA damage by gamma H2AX, and they also observe cell proliferation with a Ki67 marker. And to the results, these are the results for the first two compounds which has no albumin binder in the structure. And this, you can see the highest accumulation are in the kidneys, of course, because the compounds are excreted via kidneys. But it is also interesting that the accumulation in the tumor is also quite nice compared to all other tumors except spleen, let's say. So we have quite clear and targeted biodistribution profile. We are counting also tumor-to-blood ratio, which is expressing the suitability or the specificity of the accumulation in the tumor. So the higher this number is, the better is the outcome of the study. So when we check our number for our compound is 17, and the clinically tested PSMA has only 8.5, so we are even better in this time point than clinically tested drug. And when we watch to the later time points, like 24 hours post-injection, we see that we have even much higher numbers, of course. Because with time, the accumulation or the percentage in the blood is, of course, decreasing, and in the tumor, it's increasing. So these are the numbers for the albumin binding versions of our PSMA inhibitors, and here we can see that the accumulation in the tumor is quite high, in this case even higher, and it's clearly increasing with the time. So we can say that the idea about slowing down the pharmacokinetics works, and it leads to the higher accumulation in the tumor. And when we check the tumor-to-blood ratio, we see that the number is extremely high. So it could be very successful drug in this case. Just very briefly about the histological examination. Here on the left, you can see that there were almost no necrosis in the tumor tissue. The PSMA expression was really high, nice. But what is the most interesting is DNA damage. Here in this graph, compared to the control, in the treated mice, we have observed increase of the DNA damage. And here on the lower graph, you can see that the cell proliferation was, compared to the control, decreased. So we can say that after only one single dose of the actinium PSMA, we observe such nice effect in the tumor tissue. So let me conclude. We have tested 4 novel inhibitors of PSMA labeled with actinium-225. The biodistribution pattern of these ligands is very promising because of the high accumulation in the tumors, because of very high tumor-to-blood ratios. But we can also state that the uptake in the kidneys and spleen is also very high, so it could be a dose-limiting organs. And histological analysis shows us that DNA damage is increased in the tumors after the application of this drug, and the cell proliferation is decreased, which is also promising, of course. Our future plans are to publish these results soon, because these 2 compounds weren't published yet, and of course to perform new well-designed therapeutic study in the tumor mice. So I would like to thank all my colleagues in Olomouc and in Dresden too, grant agencies for the funding, and you for the attention.
Thank you for a very nice presentation, and we have time for a few questions. Please.
Yeah, thank you for the wonderful talk and amazing and very loud, but okay. So thank you for your amazing talk and really promising results. And I have a few questions. I was wondering in the beginning, you mentioned that the PSMA has also diagnostic potential and that it is 10 times increased. But I was wondering at what stage of the tumor or what size of the tumor you see such a high increase in the expression, because I don't expect it to be so high from the beginning, or am I wrong?
So you are asking about the clinical question about the expression during the disease, if I understand correctly.
Yes.
So,
Yeah.
I think it is quite high since the beginning of the disease, but as I'm not a clinician, I'm not dealing with the early diagnostics. I'm not sure about the exact level, but I think it's quite high from the very beginning.
Yes, thank you. And regarding the quite high accumulation in the kidneys, I was wondering whether you also checked the histological status of the kidney, whether there's any chance of nephrotoxicity or any damage which can be caused due to such a high accumulation of the PSMA kidney.
Yeah, it's a good question. I haven't shown the results from the kidneys, but we have examined the kidneys and we haven't observed any signs of necrosis or DNA damage in the kidneys. And as I saw in the clinical results from already published clinical trials, there is no real danger about the kidney toxicity.
Okay, thank you so much for your answers.
You're welcome.
Thank you for this question, and other questions. If not, thank you, Zbigniew, for your nice presentation, and I would give my microphone to Milan.
So now I would like to invite Ermin Shadeh, and he will be talking about anti-SARS-CoV-2 activity of one existing anti-inflammatory drug. So Ermin, the floor is yours.
So I will give the talk on activity of I will give the talk on activity of fenretinide against SARS-CoV-2 virus. Okay. As a morning update on the World Meter website, the coronavirus disease, COVID-19, still remains as the global problem with almost 20 million active cases across the world. Prophylactic and therapeutic measures against its etiological agent, SARS-CoV-2 virus, are still limited, and many scientific studies concern the therapy against SARS-CoV-2 virus. Different strategies were used during drug discovery. One strategy is drug repurposing, which is based on the use of pre-existing drugs. One such pre-existing drug is fenretinide, which is 4-hydroxyphenylretinamide with structure similar to vitamin A. It is a compound with anti-inflammatory, anti-cancer, and antiviral activity. This compound is interesting for us because it can change ceramide profiles that are important for life cycle of SARS-CoV-2 virus.
Yeah, this one. Yeah? You can turn off the projector by this button. Be aware. Yeah, this button. Yeah, just this. No, you have to be found. You press this, and this will shut down the projector. Yeah, be careful. No. This, only this. Yeah, you are pressing both, but only.
Okay.
there,
Okay.
yeah?
Because I have one skin which is quite something like what is called in Czech muzzle, and it presses both buttons because of pipetting. So the SARS-CoV, the key event in the life cycle of SARS-CoV-2 virus is viral entry into the target cells. There are two routes of entry. One is endocytosis and the other one is membrane fusion. The key mediator in both events is a structural spike S protein. This protein needs to be primed before interaction with angiotensin converting enzyme. So the routes differ in the enzymes used for priming of spike protein. Initially, the protein is cut by furin at the site which connects two subunits. Then in the membrane fusion route, it is proteolytically cut in the S2 unit by transmembrane peptidase serine protease, while in the endocytosis pathway, it is proteolytically cleaved by cathepsin L. Although virus can enter the cells by both routes, the entrance to the cell is occurring, according to the recent studies, mainly through almost in 70% of cases by endocytosis. So in addition to viral entry, the other events are important for replication of virus and its release from infected cells resembling endocytotic pathway. So on this slide you can see more the structure of virus, which the virus itself is belongs to Coronaviridae family. It is positive sense single-stranded RNA virus with 4 structural proteins important for life cycle. After internalization into the cell from the early endosome, genetic material is released. In the genome there are 14 open reading frames, and from the 2' end there are 2 open reading frames, and translated non-structural proteins are important for formation of replication and transcription complexes, which are associated with viral replication organelles, important for transcription of genomic RNA. Then translated structural proteins are transported to endoplasmatic reticulum, and then genomic material and structural proteins are assembled into viral particles in one intermediate compartment called endoplasmatic reticulum Golgi intermediate compartment, and which enters late endosome pathways and released by exocytosis. Viral cycle depends on the membrane trafficking and is influenced by ceramide profiles. Ceramides are lipids composed of long chain amino alcohols called sphingosine and fatty acid. They are synthesized in the endoplasmatic reticulum in the pathway which starts from palmitoyl coenzyme A and amino acid serine by serine palmitoyl coenzyme A transferase. Then sphingosine is synthesized, and upon addition of fatty acid to sphingosine, the dihydroceramide are formed, and finally ceramide are formed from dihydroceramide by desaturation, which is mediated by dihydroceramide desaturase. According to the size of their fatty, according to the length of their fatty acids, they are classified as short chain, long chain, and very long chain ceramides. They are transported to Golgi by transporter proteins, and they are intermediates for all other sphingolipids, including sphingomyelins, glucosaceramides, and ceramide phosphates. They enter catabolic pathway in lysosome, where they are degraded into sphingosine and fatty acid. So their turnover is the turnover of ceramides is is not simply influenced by de novo synthetic pathway, but also huge network of salvage pathways of other sphingolipids. So the sites of subcellular localization of ceramides coincides with the sites of important for life cycle of SARS-CoV-2 virus. The modulation of ceramides could be used to preclude a life cycle of SARS-CoV-2 virus. Fenretinide can inhibit synthesis of ceramides by inhibition of enzyme dihydroceramide desaturase, but also it can affect their signature by inhibition of enzyme stearoyl coenzyme A desaturase, which is important for profile of their fatty acids. Also, fenretinide can have other effects on other proteins and can inhibit directly even some viral polymerases. So we are focused on this fenretinide, and the objectives of studies are to determine the activity of fenretinide against SARS-CoV-2 virus, and to determine its effects against its effects on the ceramide profiles of infected verocells. The effects of fenretinide against SARS-CoV-2 virus are determined in the antiviral assays. In these antiviral assays, the cells were first treated by fenretinide, infected by virus, and treated by fenretinide for 72 hours. The results showed that fenretinide inhibited SARS-CoV-2 virus in dose-dependent manner. The IC50 was against the IC50 against SARS-CoV virus was 4.5 micromolar concentration. In subsequent studies, we determined the effects of fenretinide on viral replication and ceramide profiles in infected cells. According to the treatments, the cells are allocated to 4 different treatment groups together with 2 controls, as shown by the scheme on the slide. The effects of when the effects on fenretinide on replication level of SARS-CoV-2 virus were determined, the cells were pre-treated by fenretinide, infected, and then treated by fenretinide for 24 hours. both supernatants and both culture supernatants and cells itself were used for analysis. The viruses were extracted from the cells by freeze-thaw method. The results showed that pre-treatment and the treatment of the cells by phenretinide significantly reduced the level of infection in Vero cells. That level of infection, there was discrepancy between the effects of remdesivir, which is specialized for inhibition of replication of virus, and the effects of phenretinide, which suggest different mode of actions. So in the subsequent studies, when the ceramide profiles are analyzed, the cells were subjected to treatment as described, and culture supernatants were used to determine infection level, while the cells were used for extraction of lipids. So as shown on the sides, the infection level of supernatants resembles to those ones shown on the previous slides. This was a checkpoint, and then the studies proceeded with the analysis of ceramides. When the ceramide profiles of the cells allocated to the different treatments were analyzed by liquid chromatography, mass spectrometry, marked differences were found among them. In the non-infected cells, the pre-treatment and treatment by phenretinide induced the increase in C14, C17, and C18, while it induced decrease in the level decrease in the level in the C16-1 to C16 ratio and C18-1 to C18 ratio. Such ceramide profile is typical to phenretinide signature, which was found in previous studies. When in the cells which were not treated but infected by virus, almost all ceramide ratios were increased except C17-1. This might be due to the fact, this is quite new finding, this might be due to the fact that S1 subunit of spike protein is heavily palmitoylated, that uses quite a lot of C16 fatty acid. And the effects of pre-treatment and treatment by phenretinide in infected cells have some effects which are intermediate between the signatures of between the ceramide signatures of SARS-CoV-2 virus and phenretinide. When the very long chain ceramides were analyzed by liquid chromatography, mass spectrometry, marked differences were also found. In the cells that were non-treated, the SARS-CoV virus induced a tremendous decrease in the C24 to C20 ceramide ratio, while in the infected and non-infected cells that were treated, pre-treated and or treated by phenretinide, the C24 to C20 and C26 to C20 ratio were significantly increased. So in conclusions, I can say that phenretinide is having activity against SARS-CoV-2 virus. The virus induced typical ceramide signature in infected, typical long chain ceramide and very long chain ceramide signature in the infected Vero cells, and these two signatures in the infected cells could be modulated by pre-treatment or treatment by phenretinide. So, and it is acknowledgement, and that's the end.
So thank you very much for your very interesting talk, and I am opening the discussion. Okay.
Thank you. And I was wondering, I guess the aim of this study was to identify a potential treatment for COVID, which I think seems really promising. But I was wondering that you eventually saw the biggest effect or the best results if there was the pre-treatment and then the infection and the treatment. So do you think it would be somehow possible to introduce that into the clinical practice?
I thank you because you were listening the talk. The thing is, that's the key point of the studies. Without pretreatment, the effect is not so strong. So, and also it is whether it could be induced in the clinical studies. I don't know, but the clinical studies on this compound is ongoing at the moment. So depending on pharmacodynamic properties, on all those safety issues associated with the drug, I don't see the point why it couldn't be. In any way, I think the treatments are long lasting. So I can say from my point, yes.
Perfect, thank you. And the last question, I noticed that with the C26-C20 ratio, or even all the other longer carbohydrates, you saw quite increase even in the pre-treated and treated group without the infection. Would you expect that even without the virus infection there was such an increase in comparison to the control?
That is something. This is. Probably the fact that the virus uses more like C16. So in that case, in the second set from C20 to C26, the phenretinide can provide more effects because it is not hampered by that level of C16. So this is not what we expected, but I think that would be explanation, because I didn't know what will happen before these studies in these ceramide, very long chain ceramide profiles. But that's one good observation. The other one is that tremendous decrease in the ratio C24 to C20.
Thank you.
Just short comment also to your question that phenretinide is used in the clinical trial in the cystic fibrosis patients, and this group is risky for the COVID. Also, this group is indicated for the application of the therapeutic monoclonal antibodies, so it's really a risky group, and these people could use phenretinide really long term, so it can be also useful in the preventive of the infection. And on the the concept of the repurposing of the interesting drugs is really important. On the 11 these months there was published a new study in the PLOS ONE about the disulfiram antabus, which is retrospectively focused and evaluating veterans which are on the disulfiram, the former members of the US Army, and there is very nice correlation between the prevention of the COVID and also the outcome. No one who was on the disulfiram died, yeah, from the group, despite of the risks, predictive risks. Yeah, so I think the strategy is quite interesting and important nowadays when we would like to facilitate introduction of the drugs on the market as soon as possible in such pandemic situation.
Okay, is there any other questions? We are a little bit back of time, so I'm going to give you the microphone.
Yeah. Thank you, Ermin, for nice, interesting talk. And I would like to welcome Sonia Gurska, and she will present us the results of the screening of the HTS facility and also the importance of the evaluation of such data in the correct consequences.
Thank you very much for introduction. Good afternoon, ladies and gentlemen. As all of us know, drug development pathway is a very long and exhaustive process. It starts with basic research, which is followed by preclinical testing, clinical trials, and when the compound is successful, we finally reach regulatory approval. At the very beginning in drug discovery, at first we have to identify novel druggable target, validate this target, develop the assay which allow us to measure the effect of compound on the target, implement the assay into HTS platform, and test chemical libraries, and finally identify leading compounds or hits. High throughput screening in this step significantly accelerate because in this process we can screen large chemical libraries. As I already mentioned, in high throughput screening, we can do target-based drug discovery, but also very popular is phenotypic screening when we are selecting compounds which have a desired effect on the phenotype. Very common phenotypic screening represents cell viability and proliferation assays. These assays are very convenient, cost-effective, and can predict the toxic potential of new chemical entities and anti-proliferative potency of the compound. In our facility, we routinely used MTS assay as a cytotoxicity assay, and all compounds, the cytotoxicity of all compounds is tested on a panel of 10 cell lines, 2 non-cancer cell lines, and 8 cancer cell lines with different origin. Each plate after measurement in software, for each plate in the software we calculate the Z factor, which tell us whether the assay runs properly, whether is suitable for the HTS assay. To have a successful screening campaign, we have to have a compound library in a good quality. Here you can see the libraries we have in our institute, and all results I will present are compounds from our proprietary library, which contains compounds synthesized by our colleagues or collaborators, and thanks to them our proprietary library is continuously increasing. In cytotoxicity profiling, we have in the first step in primary screen, all compounds are tested at one concentration, and we calculate the percentage of inhibition, PI value. And when this PI value is higher than 50, it means that the compound kills more than 50% of cell population. We evaluate this compound as active. From a recent screening, you can see that from over 40 compounds, we have more than 50% compounds active. And on the bottom in this chart, you can see the ratio of active and inactive compound for each cell line. In the next step, secondary screen, active compounds are tested across 7 concentrations, and in the software Dotmatics, IC50 value is calculated. Based on this value, we can divide the compound according their cytotoxicity, for example, into these 4 groups. And routinely in our workflow, all compounds with IC50 lower than 10 micromolar in SEM cells, for these compounds we do cell cycle analysis. So let me start with the results, and at first I decided to show you the triterpenes and their derivatives. Triterpenes are natural products of plants, animals and fungi and have a broad spectrum of protective effects. Well-known betulinic acid especially has anti-HIV and anti-malaric property, but what is interesting, this compound is not very cytotoxic, and goal or the idea of the medicinal chemist is to prepare, to synthesize derivatives of betulinic acid with higher cytotoxicity effect, with higher cell activity, more stable and more soluble, to have derivatives of betulinic acid as promising anti-cancer drug candidates. In lab of our colleague Milan Urban, they synthesized lots of triterpenoids, and here you can see the results from testing of around 100 compounds. In this slide you can see the distribution of compounds according their cytotoxicity, and you can see that mostly all compounds has moderate or very weak cytotoxic activity in cancer cell line as well as in non-cancer cell line, except one cell line, SEM cells, which are most sensitive to these compounds. Here I select some examples. At first compound 23245, for example, is a derivative of oleanolic acid. And you can see that IC50 value in most cancer cell lines is between 2 and 1 micromolar. In non-cancer cell lines, the activity is, cytotoxic activity is on moderate, and most sensitive cell lines again are SEMs, and we can see this compound is quite selective for this cell line. The other 2 compounds, which are derivatives of betulinic acid and oxobetulinic acid, have IC50 below 10 micromolar or 1 micromolar in cancer cell line, but we see the cytotoxic effect also in non-cancer cell lines. So these compounds, by the modification, lost the selectivity. By cell cycle analysis, we can say that compounds increase the number of apoptotic cells, especially in concentration 5 times IC50, and we can also see the inhibition of DNA and RNA synthesis. Other group of compounds I would like to present are derivatives of nucleobases, nucleosides and nucleotides. These chemically modified analogs of natural nucleosides are in drug classification named as antimetabolites, and they still play a crucial role in anti-cancer and antiviral chemotherapy. Generally, these analogs, antimetabolites, are incorporated into DNA, and finally they inhibit the DNA polymerase or block the essential enzyme for DNA synthesis. And on a phenotypic level, we can see that these antimetabolites caused cell cycle arrest in S phase. Here are results for analogs of nucleobases and nucleosides synthesized in a laboratory of Professor Hocek. Again, it's a distribution of all compounds according their cytotoxicity, and it's clearly see the difference between cancer and non-cancer cell lines. In non-cancer cell lines, we have only few compounds with moderate, and in MRCC5 cell line, few compounds with IC50 below 10 micromolar. But you can see here in cancer cell lines, lot of compounds have IC50 below 10 micromolar, and we have compounds which have IC50 below 1 micromolar. So some examples. So at first, here you can see the analogs of 7-diazopurine nucleosides. These two compounds induce strong cytotoxic effect in cancer cell lines. Mostly in all cancer cell lines, the IC50 is below 1 micromolar, and in non-cancer cell lines, we didn't see any cytotoxic effect. But after cell cycle analysis, here we can see that 5 times IC50 concentration induces in both cases slightly increase in number of apoptotic cells. But what we didn't see, we didn't observe the changes in number of cells in S phase. But what is interesting, we found slightly increased number of cells in G2M phase. And we see that some cells are not allowed to enter mitosis. This phenotype, cell accumulation in G2M phase and block to enter mitosis is characterized it's a property of these compounds, dinaciclib, roscovitin, AT7519. These 3 compounds are known that they block CDK2, 5, 7, and 9 CDK kinases. And at this time, these experimental drugs are involved in several clinical trials, alone or in combination with a conventional chemotherapy. The other analogs, nucleobases analogs of the 7-diazopurine with modification on position 6 and 8. Again, very strong activity in cancer cell lines. And in some cases, on non-cancer cell lines, we didn't see effect. In some cases, yes. The first compound, but actually, in all cases here, you can see that the therapeutic index, the difference between cancer and non-cancer cells is quite high. The first compound looks very promising. You can see that in BJs and MRC5 the IC50 is over 50, but here BJ-LD and MRC5-LD the IC50 is below 1. What does it mean? Usually we plated seeded non-cancer cell lines in high density, and due to contact inhibitions, the division of cells is very limited. It's also visible on the dose response curve, where we didn't reach the 50% inhibition of cells, because we see effect only on dividing cells, and lots of cells is not dividing. But when we plated cells in a low density, we allow cells to divide, and then the dose response curve little bit change. and but still there are some cells which are not dividing because we are not reaching the 100 inhibition like we see in cancer cell lines. From cell cycle analysis, we can confirm that this compound affect cell cycle. We see the increase of apoptotic cells, but what we see is the inhibition in G2M cells, the accumulation of cells in G2M phase and in M phase. It looks that this compound could act as a mitotic inhibitor. My colleague, by immunofluorescence staining, observed some disruption in mitotic spindle, but he didn't observe tubulin, that this compound somehow affect tubulin assembly, like paclitaxel or colchicine do. So we exclude this statement that this compound could act as a mitotic inhibitor. But here we can see that this compound also induce increased number of polyploid cells, and this phenotype is connected with inhibitors of Aurora A or B, which this enzyme Aurora A and B is kinase which control the M phase, and when there is inhibitor, the cells, we see this population of polyploid cells. So maybe in this case we can estimate that this compound could act as an inhibitor of Aurora A or B. So finally, let me summarize that our cytotoxic assay could explore the relationship between the structure and biological activity of the molecule, as I presented on triterpenoids, derivative of triterpenoids, as well as we can estimate the mechanism of action of the compounds. Of course, it needs further investigation, as I presented on derivatives of nucleosides. And finally, cytotoxicity assay can narrow the list of our interesting, promising molecules in drug discovery. And at the end, I would like to thank to all my colleagues who contribute to this work, all our core workers, and also the grant agency for support. Thank you very much.
Thank you, Sunja, for your presentation, and we have time for one question. So if there is no question, maybe I have a comment or maybe small advertisement. I would like to advertise the talk of the Alžběta Srovnalová on the Saturday because he will talk about the phenotypic profiling of the compounds. So you can see how we can look on these data when we see the real image of the treated cells. Yeah? And it's also important aspect of the HTS. So it will somehow complement also this talk. Yeah. So thank you, Sunja, once again for nice presentation.
Thank you.
So now I'm going to introduce my colleague Peter Jubák, and he will be talking on a highly active anti-cancer drug, PNH173, its mechanism of action.
Thank you, Milan. I'm waiting on presentation, so hopefully it will open. Yes, something is here. So I would like to tell a story about the compound which is called PNH173 and focus your attention to its mechanism of action. This compound is developed in collaboration with Institute of Organic Chemistry and Biochemistry in Prague. And the structure of the compound is nucleoside-based compound, and you can see in this small table the activity, cytotoxic activity of this compound. So on the first look, you see that the activity is in 10th of the nanomoles, and on the normal cell lines, there is very nice therapeutic index. So it's not inhibiting even low density BJs and MRC5 cells. So therefore, we were quite interested in the mechanism of action. So we tried all the workflows which Sonia mentioned in previous talk. So the cell cycle analysis, also kinase profiling, and we were not successful. So there was no effect on the cell cycle, and also we tested panel about 250 kinases, and we didn't observe any inhibition. And also what we did some initial proteomic screening, and it was not giving us some conclusive results. But we didn't want it to lost. We didn't want to somehow stop our experimental work. So what we did in vivo activity in the mice models, and just to check if it's really promising also in vivo conditions. And it is really quite active compound, and you can see volume of the tumor on the upper graphs, and always below there is a survival curve, and you can see that these results are very nice. Yeah? We didn't saw previously any compound with such nice activity. So you can see prostate cancer cells, glioblastoma cell line, which is implanted into the SCID mice. Also pancreatic tumor and cervical carcinoma cell line, which is xenografted into the animals. And you can see very nice survival. And but anyway, at the time, we didn't aware of the mechanism of action of this compound. So what we did, because this structure is part of the nucleoside library, we were interested in the development of the adenosine receptor assays screening. And what we realized that our compound PNH 173 was active as an agonist for A3 receptor. So it was like first hint which was giving us some direction in the identification of the mechanism of action. Also there is a very low activity on A1 receptor, so we were focusing more and more on A3 agonist response. But as you can see, it's not very easy to identify which pathway, which downstream pathway is activated. So what we did, we tested beta-arrestin pathway, and we were not successful. There was just very, very small changes. Then we also focused on the adenyl cyclase and on the CAMP pathway, also without any positive result. What we did, we also tested another kinases, downstream kinases and pathways, and we were not successful. But on the end, we found one interesting pathway, one downstream interesting pathway, which is mTOR signaling pathway. And mTOR is involved in the regulation of the translation initiation, and also in the and the activity must be present in cancer cells. So we focus on this signaling pathway, and we observe decrease of the phosphorylation of the mTOR, then also some slight decrease of the P70S6 kinase. But most importantly, we observe the drop of the phosphorylation of the S6 ribosomal protein. So therefore, we focus on this protein more deeply, and we did also this monitoring of these activities on another cell line, so not only on the CM cell line, but also on the pancreatic cancer cell line, and we observe the same response on the PS6 ribosomal protein. And this ribosomal protein is a really important component of the mammalian 40S ribosomal subunit, and it's subject of the phosphorylation in response to multiple physiological and pathological and pharmacological stimuli. And we were lucky because we had prepared the resistant cell lines on our compound PNH 173, and you can see that we prepared cell line which is more than 700 resistant to our compound, and also another cell line K562 which is also more than 700 resistant to this active compound. So we were able to use it for the future study, and we monitor also the activity on this mTOR pathway. And interestingly, we observe the similar pattern like I was presenting on the previous slide. So you can see that the phosphorylation of the S6 protein is completely lost. Yeah, when we were looking on the PNH 73 resistant cell line, and also very similar compound PNH 192. So somehow this phosphorylation is reflecting the mechanism of the resistance in this cell line. But what we observed on the K562 was completely different. On this cell line, we didn't observe any changes in the phosphorylation of the S6 protein, and also in this mTOR pathway. So there is a high probability that another mechanism of the resistance is present. But based on these two different results, we can probably conclude that at least in the CM cell line, this mechanism is important for the resistance. Yeah. And what we did, we also did proteomic profiling on these cell lines. Maybe I would comment more the phosphorylation of the S6 protein. This protein is important for the, its phosphorylation is increased in the senescent cells. So it's marker of the, almost marker of the activity of the mTOR pathway in the senescent cells. But when the activity is lost, it's marker of the quiescent cells. So we have quiescent cells and senescent cells, and the lost activity of or lost phosphorylation of S6 is marker of the quiescent cells. So probably these cells are somehow showing the quiescent cells pattern. What we did also proteomic profiling, and what we observe that there is a significant drop of the expression of the many hundreds of the proteins. So you can see more than 600 proteins are downregulating following the treatment, and also in the resistant cells when we compare the resistant cell with the parental one. And the similar pattern is also on these pancreatic tumor cell lines. And when we look more deeply on the proteomic profile, we observe in the early intervals also some beta-catenin destruction complex, which could be associated with A3 activity. And also we observe the cluster of the ribosomal proteins which were down-regulated, and also RNA processing was heavily involved and down-regulated. We observe also down-regulation of the mTOR gene on the complex proteomic level, and after 24 hours you can see that much complex pathway now is appearing, and you can see again RNA processing, mRNA splicing, mRNA metabolic processes, and in general ribosome biogenesis. So all is somehow giving us some picture of the downregulation of the ribosomes and the proteins which are involved in the active or somehow inactivated by this compound ribosomes. The similar pattern we were observing on these pancreatic cell lines. Again, the downregulation of the mTOR signaling pathway, RNA binding, autophagy, ubiquitin ligase conjugation, and when we treat the cells with high concentration, even more express activity was observed. And here are the results of the resistant cell lines to PNH173 compared to the non-resistant counterpart. And again, we observe a significant drop of the intensities of the proteins which are involved in the RNA metabolic processes. Ribosomal proteins are downregulated. Also proteins involved in mitochondrial organization and cell cycle-related proteins, and RNA splicing is somehow downregulated. But all of these changes are somehow indicating us that the cells could be really mimicking the quiescent pattern. So the proteins are decreasing, and the cells is somehow surviving the treatment with PNH173. Of course, it's at the moment our hypothesis, and we plan to continue study more deeply these pathways and the processes in the future. And I would like to thank all the people involved in the research at our Institute of Molecular and Translation Medicine, and also our collaborators for IUCB Prague, and you for your attention. Any questions?
Thank you, Peter, and I'm opening the discussion. Are there any questions?
There was a beta-catenin degradation pathway.
Yes. So that is
the ones which is involved in the adenosine receptor signaling.
Yeah, it's interesting because we didn't observe when we directly studied this pathway by the immunohistochemistry. There were no very conclusive results, but on the proteomic level, we observe the decrease of the intensities of these proteins.
Mm-hmm.
So maybe we can try another technique to look on it again. Yeah.
For K562 cells, I think it would be good to check the sequence of the 6 ribosomal protein, because there could be a phosphomimetic mutation.
Mm-hmm, mm-hmm. Yeah, thank you.
All right. Is there any question now? If that's not the case, we are a little bit close to the discussion, and
yeah, and I will introduce the Jiří Řehulka. This is his talk focused on the synthesis of fluorinated derivatives and probably more deeply on the mechanism of action of these compounds.
Good afternoon. My presentation is focused on anti-cancer activity of 2-phenyl-2-hydroxy fluoroquinolinones. This work was a collaborative project between IMTM and Department of Organic Chemistry, Faculty of Science in Olomouc. Fluoroquinolones are organic compounds used in clinics as antibiotics. For example, ciprofloxacin inhibits bacterial strains responsible for abdominal, respiratory, and urinary tract infections. This compound inhibits topoisomerase and DNA gyrase of bacterial cells. These enzymes are involved in DNA replication of bacterial DNA. We prepared a set of derivatives of 2-phenyl-3-hydroxy quinolinone. We decided for fluor derivatives because fluorine improves or increases the probability of biological activity, and also it may improve the pharmacological profile of the compound. Actually, many drugs in the clinics contain at least one fluorine atom. So these derivatives have not been synthesized and evaluated before. So these are original compounds containing one or more fluorine atoms or nucleophilic moieties. The most interesting was compound 29 that contained also sulfur atom. Interestingly, these compounds exhibited low inhibition of topoisomerase or gyrase, but in fact we were mainly interested in their anti-cancer activity. To assess their cytotoxicity, we performed 72-hour MTSSA on the panel of cell lines derived from leukemia cells, solid tumors, or normal human fibroblasts. Out of 11 tested compounds, just one compound displayed high cytotoxic activity against all cancer cell lines. Interestingly, the non-cancer cell lines were not sensitive to the compound 29. Moreover, cell lines resistant to daunorubicin or Taxol that express drug transporters such PGP or MRP1 transporters that are associated with the most common forms of clinical resistance. Those cell lines maintain high sensitivity towards the compound 29. So we focus just on this compound. And to further explore its mode of action, we performed cell cycle profile analysis in CCRFCM cell line and found increase of mitotic marker phosphohistone H3 phosphorylated at serine 10, and also increase in G2M population of leukemia cells following treatment with 1x and 5x IC50 concentration. So here you see the lack of G1 population because all the cells remain arrested in mitotic phase, and also there is increase in polyploid cells in comparison to control. So these data suggested the compound 29 could be antimitotic or mitotic poison. Mitotics usually act through inhibition of microtubule assembly. To evaluate this possibility, I used tubulin assembly assay with pure tubulin. Known microtubule targeting agents, paclitaxel and nocodazole, were used as positive controls. Paclitaxel stabilized the polymerization of the tubulin and stabilized the microtubule filament, whereas nocodazole inhibit tubulin assembly. DMSO was used as a control. It was the solvent, just the solvent. This graph indicates that our compound, the fluor derivative 29, inhibits tubulin assembly similarly as nocodazole. To visualize this effect on cells, I used immunofluorescence and confocal microscopy. On the left, we see a nice mitotic cell with the mitotic spindle. In contrast, in positive control nocodazole and the compound 29, we see disrupted and disorganized mitotic spindle, and the chromosomes could not be aligned, so the cells are arrested in mitosis. In conclusion, from the set of new fluor derivatives, we found one compound with high anti-cancer activity, and we also resolved its mode of action. In addition, we are able to support our experimental data with in silico modeling. We have nice correlation of the measured data and the predictions, and it helps us to improve the pharmacophore of the compounds, increase their efficacy, and also it helps to improve the pharmacological profile of our compounds. These data were published in the European Journal of Medicinal Chemistry, and I would like to thank you to all collaborators and to you for the attention. Do you have any questions?
So do you have any questions? I think the output of the study is nice. Irka, do you plan any in vivo experiments? I mean, in animals?
We have, it's just, this compound is just the past, but now we have much better compounds with higher efficacy, and we have to think first about the use of this compound before we go to in vivo study.
So, if there are no other questions, I would like to thank you, Irka, and welcome here Milan with his talk focus on the triterpenes, on the derivatives of betulinic acid.
So first of all, I would like to thank the organizers to give me the opportunity to talk here about isosteric approach in the improvement of selectivity of cytotoxic analog of betulinic acid. Betulinic acid belongs to triterpenoids. It's a natural compound which is widespread in all living organisms, particularly in plants. These are mostly 30 carbon molecules, and since they don't have many functionalities, they are very lipophilic. They are secondary metabolites, so they don't occur in main metabolic pathways, but they still have wide spectrum of biological activities. We believe that the plants make them to fight with pests. In my research group, we are mostly focused on anti-cancer activity of triterpenes, and quite long ago we discovered this compound, 30-oxobetulinic acid. Actually, Sonia Gurska already talked about it, which is quite cytotoxic, but it's not selective. And its non-selectivity is quite clear. It contains a strong Michaels acceptor, which is highly reactive with pretty much anything in the living cells, so the selectivity is low. Our question was, would it be possible to improve this selectivity by isosteric approach? And therefore our goal was to replace this oxygen moiety by nitrogen moiety or CH double bond moiety, which should weaken or completely diminish the Michaels accepting part. The first part was masking these Michaels acceptor as azines. We prepared compound 1 and several or many of these organic hydrazones, and simple reaction, just condensation, gave us a library of the azines that we desired. We can see that we really obtained compounds with much better selectivity. They are active on T lymphoblastic leukemia cells. They are almost inactive in healthy cells. Unfortunately, these compounds had quite a few drawbacks. First of all, they had low stability in long-term storage, especially in water-based media. The reaction yielded just low amount of the compounds, which is also inappropriate. It was difficult to purify them, and we had lots of side products. These are some of the structures that we were able to isolate and characterize. So the second approach was to mask the oxo group by another way. This is the earlier one, and this isosteric replacement was about to replace the nitrogen with carbon, which should be more stable. This chemical reaction is quite known, straightforward, so we created some of these phosphines. They react with aldehyde pretty simply, so we obtained compound 3,1 to 351 with various substituents here in order to be able to see the influence of the substitution, because all of the substitution can change the electronic situation here and the strength of the possible acceptor moiety. Here are results from biological testing. Of course, I'm not expecting that anybody is going to read them. This is just to represent that we did it and that most of the compounds had some activity below the cutoff line, which is 50 micromole. But most of them were only moderately active. 4 compounds actually had activity below 5 micromoles. These are these 4 structures. 2 of them were selective for further evaluation. These compounds are active, stable, selective, and 2 of them were tested for the way how the cancer cell dies. This is experiment of Ivo Friedrich. He did the standard annexin and propidium iodide staining, which proves that the cells die because of apoptosis rather than necrosis, because you can see increasement of the cells in these 2 quadrants, especially at higher concentration of the compound. This is 5 times of each compound, 5 times of IC50 of each compound. This is just 1 time. Then we analyzed the DNA and RNA synthesis in cells treated by our compounds, and we can see that synthesis of both nucleic acid is inhibited by the presence of these compounds, especially again in higher concentrations. The cell cycle analysis shows that both compounds block the cell cycle in the first checkpoint between G1 and S phase. This is here and here at the concentration of 1 time IC50, and at a higher concentration the situation is a little different because the first compound inhibits it again in the first checkpoint, while the second compound inhibits it in the second checkpoint, which is at the end of S phase. This probably can be associated with other targets in metabolism of nucleic acids. Last not least, Ivo Friedrich did Western blot to show which way the apoptosis occurs, and since we have cleaved caspase 9, caspase 3 and PARP protein, we can say that it's most likely the mitochondrial way of apoptosis. And this is to the conclusion. We used to have 1 compound which was active, stable, but it was not selective. Our first attempt to improve the selectivity was successful, but we obtained azines which were unstable. At the end, after another isosteric replacement, we obtained compounds that were stable, active, and selective. All of the results were published in this article, and also Ivo Friedrich is going to have poster tomorrow, and he will show more details, mostly about the pharmacological properties of these compounds. I have to admit that we haven't finished this project completely yet, because the activity of 3 point something micromolar is not as impressive that one would like to develop anti-cancer drug with it, and we would like to improve this activity. We have synthesized some compounds based on these dienes, which are currently in nanomolar range, but I'm not going to show them because we have to study them more, and perhaps we can patent them later, and I wouldn't like to disclose these structures. So at the end, I would like to thank to all my research group. This is a picture of these wonderful people. Without them, this would be impossible. With all of the collaborators, grant support, and I would like to thank you for your kind attention.
Thank you, Milan, for your nice summarization of the results. There is a time for a few questions. So are there any? Jiří Řehulka.
I have suggestion. Milan, could you think about the assay for inhibition of ubiquitin pathway, because it's known that betulinic acid derivatives with free carboxyl at C28 and chain at carbon 30, they could inhibit the proteasome system.
actually this is good idea. You are right.
It's good that suggestion, but for you, not for him.
Okay, so we have the permission, right?
Yeah,
To make an order.
and Milan, could you please comment also the solubility of the compounds, if there are any strategies how maybe to improve them if they are not very favorable?
Yes.
Or maybe it's not the case of these compounds.
The pharmacological properties were okay at around the border of solubility, but you are right that these compounds are quite lipophilic completely. But fortunately, we still have this hydroxy group and the carboxylic group, and perhaps we can get them into some sort of inclusion delivery systems with cyclodextrin, gamma cyclodextrin. It's works that we make sodium salt of it, which is much more soluble than the free carboxylic acid. Then you add the cyclodextrin and they should be soluble in this, but we haven't tried it because, as I said, I think we will do more development in the new derivative with higher activities than.
Yeah, thank you, Milan. And I think we can welcome the last.
Who is next? Okay. Okay, and the last one is Kateřina Bendová, who is going to speak about 6-state gadolinium or nibactin for positron emission tomography imaging of bacteria from Burkholderia sepia complex, if I read it correctly.
Almost. Good afternoon. Today I would like to talk about gallium-labeled ornibactin and its use for imaging of Burkholderia cepacia complex infections. Firstly, I would like to start with some background information about Burkholderia cepacia complex. This group of gram-negative bacteria consists of more than 22 genetically distinct microbes, and the number is still rising. Although they are commonly found in environment, these bacteria can be serious cause of nosocomial infections, posing threat for immunocompromised patients, especially patients with cystic fibrosis. In cystic fibrosis patients, the infection can be quite tricky. It can be either inapparent, it may worsen the respiratory function, or it may cause dangerous cepacia syndrome, which causes necrotizing pneumonia, rapid respiratory decline, and bacteremia. The most prevalent species in these infections are Burkholderia multivorans and Burkholderia cenocepacia. To complicate the matters further, this complex is resistant to various antibiotics and disinfectants. In our work, we use siderophores, and these these low molecular weight chelators have high affinity for ferric ion. Therefore, are used by microorganisms for scavenging iron. They are produced not only by bacteria, but also by fungi and various species of plants. Bacteria from Burkholderia cepacia complex produce 4 siderophores, and these are pyochelin, cepabactin, cepaciachelin, and ornibactin. And ornibactin is produced in 3 conformations, C4, C6, and C8. And in our work, we used ornibactin C6 for specific imaging of Burkholderia infection. The first step was to successfully radio-label ornibactin, and for this we used ornibactin C6 dissolved in water, mixed it with sodium acetate for optimal pH and with gallium generator eluate. Then we measured its radiochemical purity, and as you can see, the best results were achieved with ornibactin of concentrations of 1 microliter and higher. Concerning in vitro assays, gallium ornibactin was quite stable in solutions at pH 7 and in human serum. However, in its stability in solution with competitive chelator and in solutions containing iron was quite decreased, but this outcome was expected as ornibactin was in significantly lower concentrations than both DTPA and iron. Protein binding values were quite low, around 5%, and the complex showed hydrophilic properties. As a next step, we tested gallium ornithine uptake in various pathogens. Firstly, we observed the influence of incubation time on uptake of ornithine in Burkholderia multivorans. So we incubated gallium-labeled ornithine with culture of Burkholderia multivorans, and at different time intervals, the culture was rinsed and then measured on gamma counter. And as you can see, the uptake rises in time. And for comparison, the other line shows a bacterial culture that was pre-incubated with iron. Okay, never mind. As a next step, we tested gallium ornithine uptake in various bacteria from Burkholderia cepacia complex. The highest uptake was observed in Burkholderia multivorans, while the uptake in Burkholderia cenocepacia and Burkholderia stabilis was significantly lower. The last bar shows culture number 10 that was heat inactivated before the incubation. We also tested gallium ornithine uptake in various respiratory pathogens. As you can see, there is almost no uptake in most of the strains, but the only exceptions are Staphylococcus aureus, where the uptake is slightly higher, and Pseudomonas aeruginosa, but that outcome was expected as Pseudomonas is closely related to Burkholderia. We also performed ex vivo biodistribution assay in non-infected mice. The mice were sacrificed 30 and 90 minutes after radio-labeled ornithine administration, and then they were dissected and their organs measured on gamma counter. As you can see, the complex has rapid pharmacokinetics and does not excessively accumulate in any organ and is excreted through kidneys. Here, the ex vivo results were confirmed by PET-CT images. Here you can see a mouse that was not infected, and the pictures were taken 30 and 90 minutes after the administration. The only prominent features in these pictures are site of injection, kidneys, and bladder. Moving on to muscle infection model, here you can see a mouse with its left leg infected with Burkholderia multivorans. And if we take a closer look at the CT picture on the left, there are no visible morphological changes to the tissue. But after the ornithine administration, you can see accumulation of the signal in the site of infection. The signal in the leg was not as high as we would have hoped. So given the fact that Burkholderia are opportunistic pathogens, we wondered if immunosuppression could have some impact on the intensity of the signal. And as you can see, after cyclophosphamide administration, the signal in the leg was significantly increased. We also performed quantification studies, and now the difference is much more prominent if you compare the non-infected leg to infected leg. In immunosuppressed mice, the difference is higher. We again performed ex vivo biodistribution assay, this time on infected mice. Overall, we can say that the pharmacokinetics in immunosuppressed mice is much slower, but again, if we take a closer look at the legs and compare the infected one to non-infected, the difference is much more prominent in immunosuppressed mice. And for conclusion, gallium ornithine complex has high radiochemical purity, it has favorable in vitro characteristics and high uptake in Burkholderia cepacia complex. It has optimal biodistribution in healthy mice, and in infection model it accumulates in the site of infection. For future experiments, I would like to do some more in vivo imaging and compare Burkholderia to other bacteria and to sterile inflammation. And I would also like to do a lung infection model and its imaging. I would like to thank to all my colleagues for their work and to you for your attention.
So thank you very much for your beautiful talk, and I am opening the discussion.
Okay, there are no other questions. I think the siderophores and ability of pathogens to uptake iron in the tissues is most probably associated with pathogenicity of the strains. And you see striking differences among the uptake of the siderophore and thus iron by pathogens of the same strains, by different strains of the same pathogen. So do you think it's associated with pathogenicity of individual strains, and could we test it like in the in vivo models? I think this would be particularly important because even clinically, and I'm planning work for you because we have already in the plane the Pseudomonas aeruginosa with strains with different pathogenicity, and we would like to test whether they have also a different uptake and whether uptake correlates with pathogenicity. And that would be clinically important because it would be nice to diagnose, or actually to see the most pathogenic strains primarily, because they cause the disease with highest probability. So that the two things, so there are Pseudomonas strains with defined pathogenicity to be tested for the uptake, but also the identified strains of Burgdorferia or any other pathogens which have different uptake would be nice to test for pathogenicity in vivo, whether they can actually better penetrate or cause a more serious disease in mice. Yeah, thank you.
I'm not sure it was a question, if I understood the question. Only the thing that the uptake is different according to the pathogenicity. I think it's possible. I haven't tested it yet, but I can look into that surely.
Okay, is there any other question? So thank you again, and I'm closing this session, and welcome to the coffee break.